But good morning and welcome. My name is Molly Bird and I'm a PhD student at MIT, and it's my great pleasure on behalf of New England Graduate Women in Science and Engineering, or GWiSE for short, to welcome you all here today. We are delighted to have you with us to participate, learn, and to build community during our spring summit. We are especially excited to have you all here with us in our new hybrid format. It has been a long 2 years for all of us, and we're looking forward to better days and more opportunities to connect ahead. Thank you for coming. GWiSE is an, this is really hard to read from this. Okay. GWiSE is an alliance between graduate women in STEM groups from universities across New England. We are joining together to support and celebrate women and gender minorities in STEM fields, to increase awareness of the issues we face and create change within our community. We want anyone interested in STEM to feel welcome and to be able to succeed. To make this possible, we are working to change the academic culture through events like today's. This year, our theme is building a career path in STEM, following your passions inside and outside of the lab. We are honored to have many incredible speakers with us today. Let's see. Including our keynote, Dr. Marinna Madrid, co-founder of Cellino, to share their experiences and the different career paths available in and after STEM. Prepare yourself to be challenged, excited, and inspired. A brief outline for today. Let's see. Okay. Yes, so of course we're starting off with our keynote. Then we have workshops coming up right after. Michelle will let you know which rooms you're going to, just for general directions. Then we come back together, mingle over lunch, back in the room where we had breakfast. And then we'll come back here for the panel on different career paths in STEM, and with closing remarks. And if you want to stick around, we have mocktails for a social hour. Okay. And then of course, the full program is available. Some of you have probably seen the sheets around, but we do have a QR code as well as a short link. And they should be on pieces of paper throughout. Okay. And before I hand it over to Abby, one of our event organizers, I would like to thank our hosts, GWiSE and Northeastern GWiSE, and all of our organizers who you'll see running around today that made this event possible. I'd also like to thank all of the GWiSE groups. We have their logos here, and schools that make this an incredibly supportive and empowering community. Thank you all for coming. And with that, we'll get started.
Okay, hi everyone, I'm Abby. I'm an MD PhD student at Tufts, and I'd like to introduce our keynote speaker, Dr. Marinna Madrid. Marinna is a co-founder and the chief product officer at Cellino, a venture capital-backed early-stage biotech company. She received her bachelor's in science and biophysics from UCLA after transferring from Riverside Community College. Following her bachelor's, Marinna received her PhD and master's in applied physics from Harvard University, where she co-invented laser-based intracellular delivery techniques. Marinna has been recognized for her achievements as a recipient of the Harvard Graduate Prize Fellowship and the Catalyst Accelerator Grant from Harvard Medical School. She is also on the Forbes 30 Under 30 2019 list for healthcare. I look forward to hearing her tell us more about her career journey from community college to co-founding a biotech company. Welcome, Marinna.
Thank you so much for the invitation. I'm honestly really excited and honored to be here. So I really appreciate it. And I have a set of slides, which I'll walk through, and I know there's time for questions at the end, but if any questions pop up during the slides, please don't hesitate to interrupt and ask me, because I'm super happy to have this discussion be focused on whatever is most interesting and most useful for you. Yeah.
Okay. Let's see. Does everything still look okay on Zoom? I think it should. So yeah, we'll get started.
It looks good here.
Yeah, it looks good in the room, and no one on Zoom is saying it doesn't look good.
Yeah,
so.
I've never used this view on Zoom, so it is tricky.
It looks good on Zoom. It looks good on Zoom.
Awesome. Okay. Again, happy to discuss whatever topics are most interesting and useful for you guys, but I figured I'd go through my path because
Abby mentioned, right now, I run a biotech company that I co-founded that's based off of a technology that I invented during my PhD. But I never expected to go into entrepreneurship. And I actually never even expected to go into science. So when I first graduated from high school, I grew up and went to high school in Southern California. And when I first graduated from high school, I actually moved to New York to go to NYU to study journalism. And it was a terrible fit. I hate writing. And it turns out writing is very important for journalism. Who would have known? Not 17-year-old Marina. So it was just such a terrible fit for me. I think there was also a little bit of the culture shock of growing up in Southern California and then moving to Manhattan. So I actually, in the middle of my first year, dropped out in the middle of a semester and just moved back home. And it was kind of a big decision because I had already spent quite a lot in student loans. Each semester at NYU is about $25,000. I didn't get that money back. So I moved back home and I basically just decided to start over. I got a job at like a frozen yogurt shop. I started going to my community college. And actually that's me at my community college in the top left picture, like 10 years ago. But community college was a really, really good place for me, because that was where I discovered science. And science was awesome for me because it was like the perfect mix of something that I was naturally good at and something that I enjoyed. And I had just fabulous professors. One of the things that I really like about community college is that the professors there are 100% focused on teaching. They're not also running research labs. And so what that means is they actually dedicate a ton of time to their students. And that's something that I really appreciated. And I have always wanted to, at some point, go back and be an adjunct professor, teach part-time at community college. But I had really solid professors in physics and math and biology, and I thrived there. And so they were the ones that really encouraged me to go to UCLA to pursue a bachelor degree. It's good that they encouraged me, because community college is the kind of place where it's possible to just kind of stay there and drift if you don't have a specific direction you're headed. So I ultimately decided to transfer to UCLA. And while I was there, I was studying physics. The biophysics degree at UCLA was really just a physics degree with an additional bio class, an additional biochem class. And I had heard about this professor who's doing food science. And so I reached out to her mostly for fun. It wasn't the kind of project where I thought, oh, I'm going to enter my PhD and use this project to get there. It was just that she was doing genuinely interesting work, applying science to understand different principles in cooking. And so I joined her lab, and the first project she had me working on was quantifying the probiotic bacterial content of yogurts. And so I went to Whole Foods, took a bunch of different samples of yogurts, cultured bacteria from those yogurts to figure out which yogurts actually had the highest levels and the most diverse probiotic bacteria. If you're interested, the Siggy's was the one that won over like Activia and some of the other brands that specifically claim they have the most. So I was working on this project. I enjoyed it. I thrived in the lab. I enjoyed being in the lab. It was kind of meditative to have something, physical and hands-on to work on. And at the time, my mentors were encouraging me to apply for a PhD program because I didn't have, a specific strong idea of I want to enter industry right now. I was enjoying doing science in the lab. So they encouraged me to apply for a PhD program. But what my mentor, the PI, and another woman who was a mentor just in general at UCLA for science students, they had advised me that, this project is cute, but it's not the kind of project that's going to get you into a graduate school. You need a different type of project for a graduate school application. And so my PI from that same lab put me on another project, which was a lot more fundamental, basic foundational science. And so what we were doing here was we were taking plant cells and removing the nuclei from the plant cells and essentially measuring the squishiness of the plant cell nuclei. So measuring the Young's modulus. And this was a more, basic foundational science project where we were interested in exploring this just to understand basic physics about plant cell nuclei. But there were also interesting applications for this. So for example, in cancer cells in humans, the nuclei are a little bit more squishy than nuclei in healthy cells. So you can imagine this, down the line moving into some sort of diagnostic technique. But what happened with that project is the plant cells, they have a cell wall. So they start off very cuboidal. And then if you take apart that cell wall, which you need to do to get to the nucleus inside, they become what are called protoplasts, which are these really pretty spherical shapes. And those are the chloroplasts inside, so you can see that they're green. So what I ended up doing during that work was actually inventing a technique for removing the nuclei from the plant cells. So the traditional technique involves using detergents, which is pretty harsh because detergents work by destroying that protoplast membrane. But if you leave the detergents on too long, they also destroy the nuclear membrane. And so what I did was this is a whipped cream canister. I don't know if you guys are familiar with this. So it's basically a way of making your own whipped cream at home. And the way it works is you put cream inside that canister, you pressurize it with nitrous oxide. And then when you decrease the pressure, PV equals nRT, you increase the volume of the gas bubbles. And so the cream becomes very big and fluffy. But what I did is I put the plant cells in that canister, pressurized it with nitrous dioxide, depressurized it. And what happened is it actually kind of blew up those plant cells and released the nuclei. So this was something it was a really fun project. I didn't want to spend a PhD focused on plant cell nuclei, but I learned a lot of, important concepts about how to approach work like this. I applied to graduate school. This project, got me really far in those applications, and I got accepted to a group, Eric Mazur's group at Harvard in the Applied Physics department. And so my undergrad studies were focused on physics. Eric Mazur is in the Applied Physics department, mostly working on lasers, but what had attracted me to his lab is that he also had a really strong interest in physics education, and that's unique in the physics department. But he was also doing research on how to teach physics more effectively, specifically for groups that are underrepresented in physics programs like women and minorities. And I, mentioned to you guys, I had always been interested in one day teaching at a community college. So that was a lot of my motivation for joining this group, is because I could also focus on education. And so I spent the first couple years focusing on education. We had a team and project-based physics course that I taught that I really enjoyed, but I missed being in the lab. It was just a different type of work. I enjoyed teaching, I enjoyed research. I didn't really enjoy research about teaching. It's a very different type of research than hands-on in the lab work. But luckily, his lab was an optics lab. And so there were a ton of hands-on projects available, and optics is everywhere. So they are a laser lab. Lasers have applications in basically every single field. So there's a biophotonics subgroup in that lab. It wasn't the majority of what the lab was working on, but I had a biophysics background. I was very interested in the biophotonics group. And so I actually joined the biophotonics subgroup in his lab and started working there, which is, more in line with what I'm doing now, running a biotech company. And so the project that I started working on involved using lasers to deliver cargoes into cells. And to give an example of why this could be interesting, so this man's name is Timothy Brown. In the early 2000s, he contracted HIV. A couple years later, or maybe it was the 90s, a couple years later, he developed bone marrow cancer. And he actually received a transplant for the bone marrow cancer. And what was so interesting about that process was that after receiving the transplant for the bone marrow cancer, he was cancer-free, but it also cured his HIV. And so this was a pretty interesting scientific finding. And the reason why is because you have white blood cells in your body. They have these surface receptor proteins called CCR5. HIV uses these surface receptor proteins to enter and infect white blood cells. But the donor from whom he had a bone marrow transplant actually had a rare mutation that occurs most commonly in Europeans, where they had a mutation such that their cells didn't have that surface receptor protein on them. So when they transplanted hematopoietic stem cells from that donor into Timothy Brown, those stem cells actually repopulated his entire immune system with HIV-resistant white blood cells, which is just really, really interesting, right? So for him, this is remarkable that this was doable. But taking this approach for everyone who has a similar sort of disease is just not feasible. It's difficult to get donor recipient transplants worked out at the scale that they're needed, especially for folks that are from underrepresented backgrounds. I'm half Filipino, half Mexican. I would have a really hard time finding a donor match. But what's interesting about this is that you could achieve this same effect with a personalized approach. So if you had a patient and you were to remove some of their own cells, you could gene edit them. So let's say you wanted to create CCR5 null cells, you could gene edit them to remove that surface receptor by delivering gene editing tools into those cells, and then transplant those cells back into the patient. And this is actually even better than a donor transplant because you don't risk any negative immune effects. The patient doesn't have to undergo immunosuppression. You don't have to wait for a donor to find a match. And there are also other interesting applications of delivery to patient cells. You could deliver small molecules to do drug screening. You could deliver gene editing tools or DNA or RNA to do disease modeling. So having a technique for delivering cargoes efficiently into patient cells is really important for translating this technology to where it's useful in the clinic. And so this was what I was focused on during my PhD, is figuring out how could we deliver some of these large cargoes that are not membrane permeable, not likely to get into the cells on their own, into these cells. And the answer is with lasers. So what we did was we used laser activated surfaces to deliver cargoes into cells. And the laser activated surfaces that we were primarily focused on were these laser activated gold nanopyramids. So you can see this is a schematic, and that rectangular picture is a cross section. So you can see there is that the surface coating of the pyramids is made of gold, but underneath this is actually a polymer, and then it's on a glass surface. And what's interesting is that if you culture cells directly onto those pyramids, and then cells need cell media to be happy, so they're in an aqueous environment, you can add the delivery cargo to that aqueous environment and illuminate the substrate, the laser activated gold nanopyramid substrate. Where the substrate is laser illuminated, bubbles appear. And so these bubbles actually create physical shear stress that poke holes in the cell membrane. And then the cargo that's in the surrounding cell media can just diffuse passively into the cell through those open pores. And the pores actually close up on their own on the order of tens of seconds is what we found in our research. So this is what it looks like in a schematic, in a cartoon. This is an actual image of it. So this is one of probably my favorite image that we took during our PhDs, but this is an SEM image, so scanning electron microscopy of that laser activated gold nanopyramid surface with HeLa cells, so cancer cells cultured on top. And so you can see there are many pyramids per cell. That's the ratio. And the samples that we were making were about the size of a quarter. So they were very small, but of course cells are very small. So you can actually run, large N values of experiments using these small sample sizes. And these laser activated gold nanopyramids we fabricated in the clean room. So we also did simulations of these pyramids during my PhD because we were trying to understand how they work. We knew that if we illuminated them with a laser and had cells cultured on top, we could deliver cargo to them, but we didn't understand exactly what the mechanism was. That's something we were interested in. So we simulated what happens to the pyramids if you illuminate them with the laser. And what happens is that the heat is concentrated. The heat that's generated is concentrated very strongly at the tip of the pyramid. And that's kind of interesting because if you would crank up the laser fluence, you could crank it up to the point where you could destroy the pyramids, but all the melting would just happen at the top. So it was like experimental validation that the heat was concentrated right at the top. But basically the tip of the pyramid gets hot enough to transfer enough thermal energy to the surrounding aqueous media that aqueous environment then forms a bubble. And that bubble is what physically comes into contact with the cell membrane and ruptures the cell membrane to create a pore for delivery. But it's not just nanopyramids or gold nanopyramids that do this. Really, any metallic nanostructured surface interacts with lasers this way. And so we had another surface that we made that were laser activated titanium coated nanocavities. So these again are SEM images. The image on the left is a top view. The image on the right, I actually got that by taking one of those substrates that was about the size of a quarter, and I just broke it in half manually and then scanned along the edge. And I got lucky and got this like one perfect cell that had been cut in half. And so what you can see there is since we deposited metal from the top, you actually get two metal surfaces with these titanium nanocavities. You get that continuous surface on top, which has a bunch of holes. And then you also get these discs of metal down at the bottom. And this is what these samples look like to the eye. They're on the right. These are really pretty. They're iridescent. All of the repeating nanostructured surfaces can appear really pretty and rainbowy because of diffraction. And so we did a similar thing here where we cultured cells on top of those nanocavities and imaged them with SEM. You'll notice the nanocavities are not like zoomed out. They're not as pretty or as repetitive a structure as the laser activated gold pyramids. That's because they were fabricated in completely different ways. So the laser activated gold pyramids required a significant amount of time in the clean room to fabricate. They required photolithography. These nanocavities you could basically make in your kitchen with an oven and like a glass of water. So that was the advantage of these, is that they're easier to fabricate. You can fabricate them with cheaper materials. The disadvantage is that they don't come out as perfectly repetitive, that structure. And I mentioned we were using these to deliver cargoes into cells. So in the early days, we were delivering fluorescent dyes. Fluorescent dyes, in this case, are not really biologically meaningful, like it would be to deliver CRISPR-Cas9 or DNA or RNA, but it was a good proof of concept to show that we could do delivery. And so you see that A panel, not laser illuminated and laser illuminated. So that is a surface where cells have been grown on the entire surface, and we only laser illuminated the bottom surface to show that it really is the laser activation that's leading to intracellular delivery, not just growing on the pointy pyramids, for example. And then B and C, so the green is the delivery dye. So the green shows that we delivered cargo to those cells. The pink is a viability dye. So what it shows is that even though we poked these cells with laser activated bubbles, the cells maintained viability and maintained health. And of course, we published a couple papers on this. I had actually, one first author paper from my PhD, but a number of papers, second author or third author for the biophotonics group. Yeah, so now I'll talk about what I'm doing right now, which is leading the biotech company that I co-founded. So we incorporated this company in 2017, which was when I was still in my PhD. And basically, as soon as we got funding, I wrapped up the PhD as quickly as I possibly could, which was about 3 months later. So I started working full-time at Cellino in early 2018, basically 3 months after we got funding. And Cellino was based on that laser delivery technology. So Boston is a really special place because there's a lot of really interesting science that's going on here, especially in biology. And so we were collaborating with those laser-based intracellular delivery techniques with a couple of big names in the biology field. So we were collaborating with George Church, who's really well known in genomics, also a very strong startup entrepreneur, has started multiple companies. We were collaborating with Derek Rossi, who is the co-founder of Moderna, which wasn't as big at the time, but he was very entrepreneurial minded. And so he was actually the one that told us during our collaborations, you should try to commercialize this technology. And it was interesting for us because my co-founder, Nabiha, who I'll talk about in a little bit, and we had never planned to be entrepreneurs. And so when she asked me, she was like, I wouldn't want to do this startup without you. Would you be interested? I know none of us have ever talked about doing a startup before. I was basically like, yeah, why not? If we don't like it, we can quit after a year. Like, it seemed very low stakes at the time. And so we decided to do it. And there has been so much momentum. And I'm so grateful that we took that risk because it's been a really, really fun process. And it's very enjoyable. And I do believe that what we're doing is really impactful for the world. So to give you a sense of what we're doing, our work is very multidisciplinary. So the technical team is almost evenly split in thirds between software and machine learning, laser physics and automation, and stem cell biology. And so the first step for us was really to build a founding team. So this is me, Nabiha in the middle, Matthias on the right. Nabiha and I did our PhD studies together. So we've been working together for almost a decade. So we had a really strong working relationship going into building the startup. And then Matthias, we pulled on board in the very early days. And we have a really strong founding team. And part of my philosophy for who I decide to work with or who we hire is I always want to work with people that are smarter than me in some aspect, because that's the best way to learn. And I think we've done a really good job of that with the co-founding team and with building out the team at Cellino. So we had a really strong founding team. And in the early days, our first investor was the Engine, and they've continued to support us. But the Engine is a venture fund that's based out of MIT. They're really interesting because they invest specifically in tough tech. So technology that requires significant, hardware development as opposed to, say, an app. And they were our first investor. We actually came into contact with them kind of serendipitously because we were looking for lab space, and there's Lab Central, but Lab Central is very bio-focused. And so we recognized that we needed something more multidisciplinary. You need specific lab space to be able to run a laser. We needed a fabrication space to make the laser activated surfaces. And so we went to the Engine and we asked them about renting out their lab space. And they said, oh, actually, the companies that live in this space are our portfolio companies. And I was like, oh, what fraction of them are the portfolio companies, thinking we could just rent space? And they said, at this point, 100%. And then they ended up calling us back in a week or so and expressing interest in investing. So it worked out really well. They were our first investor. We've had a couple rounds since then, and they've continued to support us. We are still at the Engine today, which is a really nice location. It's great for attracting talent. So they've been a strong community for us from the beginning. I mentioned that the technology that we're building is very convergent. So the technology started off in a very different form in our PhD lab. We were using lasers to deliver cargoes into cells. What we're doing now has evolved quite a lot since then, and I'll talk a little bit about that. But we are still using lasers to manipulate cells. But instead of deciding by eye, I want to deliver cargo to these cells, we use software and image-guided machine learning algorithms to characterize cells and decide which cells to manipulate with the laser. And there's also a lot of stem cell biology that's involved. So we're running stem cell biology processes like reprogramming and differentiation that we never did in our PhDs. And so this is an image of the automated R&D work cell that's running right now at the Engine. This is a really nice system because it can take images, for example, in the middle of the night without a biologist having to go in to do it. So we're able to image cells, for example, every 8 hours and collect a lot of data. And it's well plate based. So you see that robotic track in the middle, in the right? That robotic track takes well plates and moves them automatically from subsystem to subsystem. So it moves it from the incubator to the automated imager to the automated laser processor to the liquid handler and back. And I know, in stem cell biology, there's this culture of being expected to go in on the weekends. A lot of scientists will go in, just look at their cells under the microscope every single day because you kind of need to check them daily to understand how they're doing. This system automatically images the cells without any scientists having to go in, and those images are uploaded to the cloud and then downloaded onto Slack. I don't know if anyone here uses Slack, but basically one of our stem cell scientists was telling me recently that she will be like on the tee and she'll just scan through the images that have been taken recently of the cells, and she can say already from those images like, oh, this cell is doing well, or this batch of cells is not doing well. So that's a really nice system to have. And what we're doing here is we're using this automated system to develop image-guided machine learning algorithms, develop laser protocols to generate cells that we want. But our big goal is to move into therapeutics. So in parallel to running this automated development system, we're also designing a clinical grade system that incorporates image-guided machine learning and laser processing. And the goal with that system is to generate stem cell-derived cell therapies for patients suffering from chronic degenerative diseases. And the idea there is that there are some patients that are suffering from certain diseases because the cells in your body die off over time. So for example, Parkinson's is one of the most well-known ones. Dopaminergic neurons die off in your brain and your body doesn't regenerate them. So one area that we're working on is essentially taking skin cells or blood cells from a patient, reprogramming them into stem cells, and then differentiating them into dopaminergic neurons so that we can then transplant those dopaminergic neurons back into the patient to treat Parkinson's. And so, I've talked a lot about how we did intracellular delivery during the PhD, and what we're doing now at Cellino has evolved quite a bit. And it's something that I like to mention because building a company or a startup out of a PhD technology kind of felt like doing things backwards. Like the right way to build a company would be you identify a problem, figure out the best solution for that specific problem, and then build a company around it. But what we had done is we had a solution that we were already building in the lab, and we were trying to backfit it to the right problem. And as applied physicists, you just don't know what is the right problem for the biology field. You really have to talk to people and listen to people to figure out what that problem is. And so the first couple years of Cellino, I would say, were all about figuring out what is actually the right product market fit and having to put our own inventor scientist bias to the side because we had our own opinions about what we thought was most interesting to do with the technology, but that doesn't necessarily match what the industry needs. And so the first couple years for me and Nabiha, the majority of the work that we were doing was going out and talking to folks in biology and medicine and the regenerative medicine space, discussing our technology and asking them, like, what problems do you have that we could solve efficiently with this technology? And it was funny because a lot of folks asked us, like, well, could you use the laser to kill cells? And we were like, yeah, that's like the easiest thing we can do with the laser. Delivering cargoes into cells is way harder because you have to get exactly the right laser fluence. If you go too high, you kill the cells. If you go too low, you don't get delivery. So like there's a very narrow range where you can deliver cargo successfully. But to kill cells, you just have to be in this threshold. You just have to be above that threshold. So it's much simpler from a laser processing perspective. And so when we were talking to folks who were in big pharma or in biotech companies, they were like, well, killing unwanted cells is a huge problem for us. Imagine you're transplanting a cell therapy into a patient and you have just 100, and cell therapy transplants include hundreds of thousands or billions of cells. That's a therapeutic dose. If you have just a couple hundred potentially tumorigenic cells, that is really dangerous for the patient. So there is a need to literally just kill off the unwanted cells while leaving the healthy cells behind. And that is definitely something we could do with the laser. So that's something we're doing here. This is, when it happens in real life, it's very fast on the automated system because our laser has a repetition rate of 200,000 pulses per second. But here we've slowed it down so that you can just see it in real life. So these are induced pluripotent stem cells. You would never want to transplant induced pluripotent stem cells directly into a patient. They would be tumorigenic. You want to differentiate them to a therapeutic cell type like dopaminergic neurons and then transplant those. But some differentiation protocols are not 100% efficient. So some of them leave these induced pluripotent stem cells behind. So what you need is to be able to identify them via imaging and laser target them to kill them. And so that's what we're showing here. You can see they're glowing with the viability stain when they're alive. And then after they're laser processed, we can kill individual cells without harming the neighboring cells. And after we found the right product market fit, the company really took off. So we closed an $80 million Series A at the beginning of this year. It was co-led by Leaps by Bayer, which is a big pharma, which is huge validation for an early stage biotech company, Humboldt Fund and 8VC, with contribution again from the Engine, who has been our supporter from the beginning, Coastal Ventures and Felicis Ventures. And so what we're using that funding for is really to build the first platform that's using image-guided machine learning to characterize cells, laser manipulation to process cells for scalable cell therapy manufacturing. And what's unique about the way we're doing it compared to how others are doing it is that because this is so scalable and this approach is so cost effective, we can generate personalized cell therapies, meaning from a patient's own cells in a cost effective way, which just hasn't really been done before. And that's especially nice for specific groups like underrepresented minorities that would have a really hard time finding a donor match for a transplant. And so this is my last or second to last slide. There's a lot that I am happy to discuss in terms of making that transition from academia to industry. The main point of advice that is really useful for everyone, no matter whether you're staying in academia or not, is just relying on mentors and networking heavily. And that's something that I have definitely relied very heavily on my mentors. Networking is something that I didn't always enjoy, that I've learned to enjoy, but it's necessary. In industry compared to academia, so when I was in my PhD, we were doing simulations to understand how the bubbles formed. We were doing a lot of exploratory science, and we prioritized projects based on what we would learn the most from or what was most interesting. And that's very different from how work is done in industry because there isn't as much focus on how the science works. All of the efforts are really focused on just building a product, de-risking that product, and the timelines are really efficient. But I've kind of enjoyed that difference in pace. I think there's a lot of momentum in what we're doing. I've really enjoyed that aspect of being in a startup and being in industry. In part 3, one piece that was really difficult for me in the early days of transitioning to the startup is that there are a lot of really big, important decisions that had to be made without enough data. So my comfort zone is collect a lot of data, make a very rational, logical decision based on the data and a decision that I feel confident in. But in a company, it's actually more important to make a decision quickly and have the entire team be working and moving forward on that decision and then pivot later if it's the wrong decision. But the worst case scenario is we wait to collect a lot of data and don't make any progress in that time period. Because even if you've made the wrong decision, you've at least made some progress, you've made some learnings from that, and it's kind of what you need to do to move things forward. And there's just a lot of decisions that you'll have to make where you'll never have all of the data. So that was something where I really had to push myself out of my comfort zone to get to the point where I could do that efficiently. Number 4 is a question that I get a lot from my friends that are in their PhD program and entering their first job, whether or not they should negotiate the offer they've received. And the answer is yes, 100%. You should absolutely always negotiate. If you are getting an offer from a company, they are offering you something that is in the lower range of what they can offer you because they expect you to negotiate. So if you don't negotiate, then you're missing a really big opportunity. And I can talk about best practices for negotiation. It's you should never be the person to give the first number. You should always ask them to do it. If they ask you in an interview, you should deflect, and there are things you can say. I find it's a lot more comfortable to negotiate via email than in person or over Zoom or over the phone. That way you don't have to deal with people's like first reactions or gut reactions to a specific number. And then the other point I'd like to make there is that the negotiation is for an entire package. So there's the salary, which is what most people focus on, but there's also stocks in the company. If you're being offered stocks, you should pay close attention to whether those are options or shares, because that's very different. One is they're giving you stocks, shares that you could sell and make money from. The other is they're giving you options, which means like an opportunity to buy a stock with the hope that it increases in value later. And then there's also benefits and time off, and there's some flexibility, but the negotiation process is a process that has a lot of levers, and you can usually push on one if you're willing to give up a little bit on the other, or sometimes you can push on multiple. You can also negotiate for title, and I view title as being most important for where you want to go in the next phase of your career, as opposed to that specific phase that you're being hired into. And then part 5 here, honing the soft skills. I think there are a lot of skills that were really important for industry that aren't necessarily taught in the PhD program, or if they're taught to really varying levels, just depending on that PI's particular interest and whether they care to teach those skills. And so in industry, what I found is that it's not so important like who has the most clever idea or who's the best at scientific debate, but there are a lot of like teamwork skills and communication skills that go a lot farther than just being the smartest or most clever person in the room. And so over communicating, I like to emphasize this a lot. You know, if you've said something to someone in person, following up via email or via Slack so that you have it in 2 different modes of communication. People management is its own entire skill that requires a lot of effort and a lot of learning that is just not taught at all in the PhD program. And there are really nice management courses that you can take to learn best practices on how do you give someone feedback, and at what frequency do you give someone feedback, and how do you give it in a way that it's constructive and doesn't make them feel bad and leaves them feeling motivated. And then project and meeting management. This is something that we put a lot of thought and effort into so that people don't spend all their time in meetings where they're not needed. Basically, meetings should be for discussions that require discussions or decisions to be made. Updates should not happen at meetings. Updates can happen via Slack or via email. Meetings are times where you really want people actively involved and not spending too much time in meetings so that they actually have time to work. One book that I really like for making the transition from academia to industry is called It's a Game, Not a Formula by David Giltner. So he's from the optics and photonics space. It's a really nice book. He has a lot of interviews that he's done with PhD students that have entered into different parts of industry, including big companies and the startup space. And then this last part, one of the organizers asked me to talk a little bit about my values and how they align with the career choices I've made. And it was an interesting question that I've never gotten before. But 2 of my most important values are adventure and independence. And I think it wasn't intentional, but those are values that definitely align well with startup life, if that's something that you're interested in. So like, for example, for me, it's really important to maintain my independence and be able to make my own decisions. I think if I was working at a company that was telling me you have to wear a specific suit and you have to clock in at 9 and you have to clock out at 5, I would be really unhappy, even if the people were great and the science was great. So what I like about the startup life and having started my own company is that I get to make a lot of those decisions for myself. And I don't think I work fewer hours on average. I think I end up working more hours on average than a 9 to 5, but I'm happy that someone didn't tell me to do it. If that makes sense. Like, I'll work. I just don't want to be told to work. Yeah, so those are just some general pieces of advice. Happy to talk about anything that is most interesting for you guys.